Everyone's doing well. Okay, so I'm going to go over the protein backbone and the Ramachandran diagram. So first, the protein backbone, and we're going to look at it with the goal of understanding protein folding. So protein folding is a conformational change. So you're not going to necessarily have any forming or breaking of covalent bonds, but instead we'll see to get those different conformations, you're going to have different rotational angles about those covalent bonds. And those are going to be your torsion angles, or they're also called dihedral angles. And there are 3 of them. So between your nitrogen and alpha carbon, we have phi. Between your alpha carbon and carbonyl, we have psi. And then between your carbonyl and nitrogen, we have omega. And because of this double bond character that we have here, omega is actually going to be partially restricted. So omega is restricted to 0 degrees or 180 degrees. That's your cis and trans, and that's referring to your alpha carbons. So in this drawing, the alpha carbons are on opposite sides. This is omega is 180. And the trans conformation is going to be the more common conformation. So we're just going to work under the assumption that omega is 180. And so that leaves phi and psi and variations in phi and psi to determine how your protein is going to fold. So phi and psi determine protein fold. And some combinations of phi and psi are going to be allowed and some aren't. And so we can visualize this with the Ramachandran diagram. So here we have phi, psi, and then remember omega is going to be 180. And we have your full 360 degree range of rotation. And then we have 3 outlined regions. And these are going to be regions that are allowed combinations of phi and psi. So we have alpha, beta, and L. And then the rest of this white space is forbidden conformations, because you're going to get collisions with the backbone. So you're going to have, with some combination of phi and psi, you're going to have 2 atoms closer together than their combined van der Waals radii. So this puts 2 atoms closer than their combined . Right, so you're going to get van der Waals repulsion. And you're going to have this extremely strong repulsive energy that you're not going to be able to reconcile with other stabilizing forces. And so we just say that these conformations are forbidden. And so now looking back at the allowed regions, so if I have phi and psi here in this beta region, what exactly does that mean? Well, if you just know one residue, that actually doesn't tell you that much. But rather, if we have a beta strand, then what that does tell you is that you're going to have consecutive residues with phi and psi values clustered in this beta region. So you'll have a clustering of phi and psi, and you'll have a beta strand. And then similarly, for an alpha helix, you'll see consecutive residues with clustered phi and psi values in the alpha region. And then for L, you would see like a left-handed helix, but that's not going to be very common in proteins. So alpha helices and beta strands have clustered phi and psi values. But proteins aren't only alpha helices and beta strands, right? So you're also going to have some unstructured regions. So these would be loops. And what we'll see in loops is that instead of clustering, you'll have consecutive residues with scattered phi and psi values. So you'll have one here and the next one here, here, here. So that'll get you a loop. So consecutive residues have scattered phi and psi values. And that's kind of why if you only know one residue, you're not going to be able to tell what kind of structure it's in, because it could be, you know, that it's in a beta strand or some unstructured loop. And then there are some things that I do want to point out about this diagram in particular, because there are other variations of Ramachandran diagrams. So this one is generated from alanine. So taking alanine, trying different combinations of phi and psi, seeing if there's a collision or not. But in general, it does also hold for other amino acids. And hopefully you'll be able to see why. So I have alanine and leucine, but this is going to apply to other amino acids as well. So here's your alanine side chain, your leucine side chain. It's bigger, so anything that is forbidden for alanine is also going to be forbidden for leucine. But then also notice that the side chain is growing away from the backbone. So growing So as you're rotating your bonds, you're not going to have any more collisions with the backbone. And so all the regions that are allowed for alanine are also going to be allowed for leucine. And I say in general, because of course there are exceptions. So I do have a quick poll. So I have Ramachandran diagram here. And it looks different from the one you just saw. And so my question is, which amino acid was used to generate this diagram? And so histidine, proline, or glycine. And for comparison, here's alanine. That's kind of drawn in the same style from the same paper. So take a moment to think about it, and you can just drop 1, 2, or 3 in the chat. Cool, yeah, I'm seeing a lot of 3s. Would anybody like to offer their reasoning? I would agree, I would say it's 3. Okay.
It's because glycine is the simplest amino acid, so there's less collisions possible, so there's more regions, like different kinds of conformations it can take.
Yeah. I didn't hear quite clearly, but I think you got the gist of it. So we have more allowed regions, right? So less collisions as you're rotating the bonds. And so because glycine has just a hydrogen instead of methyl, we will see more regions that are allowed. Proline, actually, you'll see that it's more restricted in the conformations that it can take on, because the side chain is attached to the backbone. So that's going to introduce some additional strain as you're rotating your bonds. And so some conformations that are allowed for alanine might be too high energy to be allowed for proline. Okay. And then the last thing that I wanted to point out is that the earlier diagrams, they're all assuming that atoms are hard spheres. And so this produces a binary diagram. Right, so we either have allowed or not allowed, right? So you either have no collision, no collision, or you have collision. So it's purely based on like comparison with your van der Waals radii. But as you know, there are other energy terms besides van der Waals. And so you also have hydrogen bonds, ionic interactions, interactions with water that could provide some degree of stabilization. And then moreover, there are your molecules aren't at fixed energy. So at any given point in time, they'll have like a certain probability of having a certain energy. Um, and that probability is given by the Boltzmann distribution. Right, so some molecules will have higher energy than others. And so if we take into consideration these two things, we can get a diagram that looks more like this. And so this is a lot more detailed. It's not binary. You still have your alpha region, beta, L. So these are low energy, and we have high probability of these conformations. And then with the higher energy conformations, you'll have low probability. Oh, probability. So it's not as clear cut as like, yes, you can have this conformation, no, you can't have this conformation. But as you do increase in energy, those conformations are going to be exponentially less probable because of the Boltzmann equation. Okay, that's all from me, but are there any questions? And if not, I will pass it off to the next tutor. But I feel free to send me questions in the chat as well.
I'll share my screen.
Madeline, you're kind of echoey, so you might want to mute one of yourselves, maybe.
I only have one of myself.
Oh, interesting. Okay.
I'm not sure. I hope it doesn't.
Okay, that sounds better. I don't know why I was echoey.
Okay, let me share my screen. So, as we have already talked about, we've discussed some of proteins' secondary structures, alpha helices, and beta sheets or beta strands. And we've also discussed tertiary structure, where we have the overall protein fold. And we have this formation of a hydrophobic core, and that this drives protein folding. But now I'm going to discuss a little bit about protein domains. So a protein domain is a region of a protein structure that has a functional purpose. And there's a variety of protein domains that that span various proteins in our body. Some examples were this SH1, a tyrosine kinase, or an SH2 domain, which binds phosphorylated tyrosines, and an SH3 domain, which binds proline residues on other proteins. And these are just examples of domains that are highly conserved, and you see them in a variety of proteins. And we discussed that having more domains in your protein and mixing them up can form more complex proteins in our body. And that's just one way proteins can diversify their functions, and how more complex proteins have different regions that do different things. We also discussed structural motifs that are often found in proteins. And these are 3D arrangements of two or more secondary structural elements. and they're commonly found in proteins and are often part of larger protein domains. These, you probably don't have to know any specifics except for the coiled-coil domain, but these are just some examples. Helices connected by turns or loops or hairpins on antiparallel beta sheets. Or this is a nifty one that you can see the formation of parallel beta sheets with this alpha helix being offset from that plane. But we will focus later on the coiled-coil domain, or a different tutor will discuss in more detail this domain, or a motif, I mean. So seeing as we have a lot of, or the driving force for a lot of the protein folding is the formation of hydrophobic cores or regions. So key features of this is amphipathic or amphiphilic alpha helices and beta sheets. So this, or amphipathic, just means that one side of our alpha helix is hydrophobic and the other side is hydrophilic. And this allows for this hydrophobic side of alpha helix packed around the protein core, whatever that may be, and the other side to face water. So it's very common to see amphipathic alpha helices and beta sheets of proteins. So the same holds for a beta sheet, where one side of the plane is hydrophobic and the other is hydrophilic. This amphipathic beta sheets are commonly found in transmembrane proteins, for example, where you can have a little tube come through the membrane, and it's made up of beta sheets where the hydrophobic side faces the membrane, and the hydrophilic side allows for polar molecules to pass through the channel. So now we can discuss a little bit about how, for the structure of this amphipathicity in an alpha helix. We saw this helical wheel introduced to us, and because an alpha helix has 3.6 residues per turn, we deduce that there is 100 degrees between each residue. So if we start with residue I and we go 100 degrees around this helix, either facing up or down. In this case, we'd be facing down on the helix, moving up. We get to I+1, then we can move another 100 degrees and get to I+2, and so on until we see that I+3 and I+4 residues are on the same side of the helix. And this is an important point for amphipathicity. If we have hydrophobic residues, I+3, and I+4, they're all on the same face of our helix. And this is a pattern that we think is important. So you know that I+3, and I+4 are on the same side of the helix. So that's I+3, and I+4. They all face the same direction. And then when we look at beta sheets, it's a little simpler. Since the residues of the amino acid chain are above and below the plane, in order to have an amphipathic beta sheet, most residues below the plane be hydrophilic or hydrophobic, whichever one, and then the residues on top of the plane will be the opposite, and you have an amphipathic beta sheet. And that was pretty short. I think that wraps up what I have for amphipathic helices and sheets. If there's any questions, I can insert in the chat. If not, I will exit my share. Stop sharing.
Okay, hello everyone. So we'll be talking about protein tertiary structure for this section, and let me know if you can't see my screen. Okay.
We can see it.
Okay, thanks. Okay, so the first thing is, let's go over the main idea that isolated alpha helices, so if we just have one alpha helix, they are not very stable on their own if they form this, this structure. And that's because of the intrinsic competition that water has for the hydrogen bonds that the alpha helix is making within itself. So whether or not this, this peptide is making the hydrogen bonds with water or with itself, there's no net gain in hydrogen bonds or stability, so it's not very stable on its own. So because of this, they often seek greater stabilization when there are more alpha helices and they can have more interactions with one another, and this is why tertiary structures are important. So the first thing that we're going to be talking about is coiled coils. So they are called coiled coils because alpha helices themselves are coils, but then they also coil around one another. So that's why there are two coils within the name. Two helices, they can coil in two different directions. So they can either be parallel, meaning that, you know, the N terminus is on the same end, the C terminus is on the other end, on the same sides, or anti-parallel where they're operating in different directions. And in the textbook, it says that parallel coiled coils are more common than anti-parallel ones. And then just as we saw before, residues at the I+3, and I+7 positions face each other at the interface. So this is going to become really important when we see how these residues are interacting with one another. So the residues at these positions are usually hydrophobic, and we'll see why. The main idea for this section will be that the main driver behind coiled coil stability really lies in the hydrophobic residues. So as we saw, or as we just talked about, the hydrophilic residues they're mainly contributing hydrogen bonds, but that's not really adding anything if they weren't in this coiled structure. So instead, what the coiled coil is adding are the grammar are these hydrophobic residues that we'll see in a second. So first, I just wanted to ask a quick question and see where we are. So alpha helices are either right-handed or left-handed, and coiled coils are right or left-handed. You can just drop the number in the chat. Cool. Yeah, so I'm seeing a lot of twos. That's great. So alpha helices themselves are right-handed, and this is just intrinsic to their nature. All naturally made amino acids are right-handed or like secondary structures, but coiled coils themselves are left-handed, and we'll see why on the next slide. So they are left-handed, and this is because of the amount of residues per turn. So we know that there are 3.6 residues per turn, and if we go up to the I+7th residue, it falls short of the parallel axis. So if we're following this up from the bottom and we're going around, the I+7th residue doesn't quite make it to a full 360-degree turn. So the I+7th residue falls a little bit to the left. It would be different if the alpha helix itself was left-handed, then we would be seeing the opposite trend. But since the alpha helix is right-handed, we see that the residue falls short. Then if we want to coil these two coils together, this red one is essentially the same copy of the blue one. We just want to bring these two together. And to create some extra stability, we would imagine that these two yellow spheres are hydrophobic residues, and we want to have them interact with one another to provide some extra van der Waals stabilization, things like that. The only way for them to line up is if both of them leaned a little bit to the left so that they would coil around one another in a left-handed fashion. And because of this, supercoiling or coiled coil interactions, they reduce the residues per turn from 3.6 to 3.5. So with the 3.6 residue per turn, we see that this residue falls short. With the global change in, with the global rotation that we see, this residue will line up with the axis, so it will reduce it to 3.5, and 2 turns will perfectly align with the I+7th residue. Okay, and then moving on to the specifics of what makes this interaction happen, we have this handy dandy table over here that shows what types of interaction or what types of residues are usually at these positions. So first we can see that we have a parallel coiled coil here, and we can start with the A position. We're noticing that we're going clockwise in this scenario, which means that the N-terminus is on the top and we're looking down. And same thing for the other one. The A and D residues will be the ones that are interacting at the surface. And just keep in mind that the A and the D level, they're not like at the same level. They are stacked one on top of the other. So the A's will be interacting, and it's not like the A's and the D's will be interacting in this scenario. So the A's and D's will normally be hydrophobic. So that's to create this extra affinity for one another. And there will be charged residues on the sides, normally E and G, and those will normally be charged to set the register of how these two will interact. What that really means is to make sure that they are aligned properly. So if we have really strong interactions here with like G being plus and E being minus, then it'll faithfully return to this register. And occasionally A and D will be polar, but we'll see normally that they are hydrophobic. And it's labeled A through G just because it repeats every 7 residues. And a couple things to watch for whenever you get questions about coiled coils. Firstly, just make sure to note whether or not you're going to be labeling this clockwise or counterclockwise. You'll know this by whether or not it'll say that, oh, the N-terminus is on the top or at the bottom. And then also note if they are antiparallel or parallel. On the next slide, we'll see how these two interactions look different if it's parallel or antiparallel. So this is the parallel case. We saw that the D of one was interacting with the D of another, and then the A's are interacting. In regards to the charge residues, it was the G and the E that was interacting. So for the parallel case, the hydrophobic ones, they're the same letters interacting, but for the charged ones, it's the opposite letter. And then if we see in the antiparallel case, now we see why it's important to note whether or not it's going counterclockwise or clockwise. The left one is at the N terminus, so we would imagine it's like spiraling down, whereas for the C terminus, it'll be spiraling in the opposite direction, so this is moving counterclockwise. And now we see that the interaction pattern is a little bit different. Now with the hydrophobic residues, the A's and the D's are interacting with one another, whereas for the charged residues, the same letter is interacting. So this is either something you can memorize really quickly, or if you draw it out, then you'll always get the pattern right. So yeah, understanding the charges of the different amino acids. Let's say a G was here, was positively charged, and an E was positively charged. Then you would know that they would rather be in this case where they're not interacting if they were both positively charged, as opposed to if they were in this case, then it would create a lot of electrostatic repulsion. So that can be one way to help discriminate whether or not they will interact in a parallel way or an anti-parallel way. Okay, and then what we just studied was coiled coils, and this will happen when 2 alpha helices themselves are just isolated. But if we have more than 1 or more than 2 alpha helices, they often will form bundles. So in this case, we have 4 alpha helices, and we see that by packing together in such a fashion, they create their own little hydrophobic effect, and they bury these hydrophobic residues in the interface. So this is just another common motif that we see. So we've studied coiled coils, and now we have this like 4 alpha helix bundle. I don't know if there's an actual name. And then the last thing that we'll be talking about is the ridges into grooves pattern. So before we saw with coiled coils, there needs to be some sort of global turn, but for ridges into grooves, this happens with alpha helices that are straight and their axis is exactly straight. So alpha helical structures that don't form coiled coils will instead adopt this ridges into grooves pattern with straight helices. And the angle at which they will interact with one another is purely dictated by the geometry of the surface. It doesn't really have anything to do with what type of sequence is there. It doesn't matter what types of residues are there, unlike the coiled coil. And we'll see why in a second. So in this sort of packing, the ridges of one amino acid pack against the grooves in another. So it's sort of like this lock and key, maybe that's a bad term to use, or like sort of like a Lego fit, where one protruding interface will fit into the hole of another. And the protruding interfaces are, the ridges are formed by protruding side chains. So this here we see like a 5-membered ring, so we can sort of assume it's histidine or something, and it's fitting into a space where there is no side chain that's coming out from the other one. And here, they're sort of stabilized by van der Waals attractions. I think that might need to be adjusted. But the important thing to remember here is that it's not sequence specific for this type of packing. And here, this is an image from your textbook. A couple of things to note is that if we just follow this pattern really quickly, we have I plus the I plus 4, I plus 8. So if we have this sort of pattern that's increasing by 4 side chains, this forms a 25-degree angle. Whereas if we have I plus 3 side chains, this forms a 45-degree angle instead. And this is purely dictated by how the helix forms and the angles that it forms. And just to understand like why these interactions have to happen at a certain angle, we can take this example of helix 1, helix 2. This forms a 25-degree angle, and this forms a 25-degree angle as well. So we can assume that the ridges that are formed here are from I plus 4 side chains. So to see how these two will pack together, first we want to make sure that the two interfaces that we want to be interacting are facing one another. So first we're going to flip this helix so that this is the side that will interact with that one. And we see that now we have to line them up. If we rotate the blue one 25 degrees, it'll be perfectly straight, but that won't really align into the ridges and grooves that helix 1 is creating. So in order to compensate for that, we have to rotate another 25 degrees, which results in this total of 50 degrees that we need to rotate so that both of the helices will fit into each other's grooves. So that's how you would do one of these problems and figure out the crossing angles that they will interact in. And then a big takeaway from ridges into grooves packing is that since it doesn't since it doesn't depend on the sequence itself, it often survives big changes in sequence. However, if there is a mutation of some sort that makes it not amenable to the interaction that's being formed or the ridges into grooves packing, then that mutation will simply not survive. So these ridges are only based on the existence of side chains, not the side chains themselves. So this often underlies like a conservation of structure. We'll often see that proteins adopt very similar structures, even though their sequences are really different. And that's because this ridges into grooves packing, for example, is not dependent on these sequences and just the fact that there are side chains in general. Okay, any questions? I will stop sharing so that Rosa can go.
Before I go, just one logistical note is that we started uploading our session recordings to one singular YouTube channel, and that YouTube channel is linked in the Piazza post with all the Zoom information. So if you ever seek these out, then they're all localized or centralized, sorry. Okay, so I'm going to be talking mostly about the hydrophobic effect, and then at the end, I'll be talking about membrane proteins. So what drives protein folding? And as you probably know, hydrogen bond formation is not the dominant driver of protein folding. And why is this the case? Well, when peptide backbone to peptide backbone hydrogen bonds form, if we look at this diagram below, it depicts the formation of hydrogen bonds between different regions of the peptide backbone in order to give rise to secondary structure. Here's a beta sheet, here's an alpha helix. And what you can see is that here, on the left-hand side, you can see that initially in the unfolded state of the protein, there are backbone water hydrogen bonds. And so when you form protein secondary structure, these backbone water hydrogen bonds are just displaced. by hydrogen bonds between different regions of the backbone. So you can see here that they're showing you backbone hydrogen bonds. And so the result of that is that the change in the number of hydrogen bonds is roughly 0, or there's not much change. And so this results in not much net gain in hydrogen bonding energy. From the folding of the protein. And so another way to state this, is that there's not much additional stability in the folded protein from H bonding. Okay, so now that we've established that hydrogen bond formation is not the dominant driver of protein folding, what is? And this was already briefly touched on, and that is the hydrophobic effect. So first, we probably want to define in more general terms, what is the hydrophobic effect? And so I'm going to state it in this way. The hydrophobic effect has a consequence on protein folding, and protein folding is driven by the preference of hydrophobic side chains to pack together. And so the hydrophobic effect is this preference of hydrophobic side chains to pack together. And this results in the formation of a hydrophobic core in the protein. Okay. And so some key points that I want to make before I go into my explanation is that water-water interactions are relatively strong. And you can imagine that when I'm talking about water-water interactions, I mean hydrogen bonding between the water molecules. And in comparison, interactions between nonpolar side chains are going to be weaker. And you probably know that when I talk about interactions between nonpolar side chains, I'm mostly talking about van der Waals interactions. Okay. So here is a fun picture of a water hydrogen bonding network. And what happens when you add nonpolar molecules, so in our case, a nonpolar or hydrophobic protein side chain to an aqueous environment, so to water, for instance, this water hydrogen bonding network is disrupted. And so that's depicted in the cartoon. You can see that the hydrogen bonding network has to go around the nonpolar solute molecule. And it creates a sort of cage connected by hydrogen bonding interactions. And I think this is sometimes called a clathrate, but I don't think that's important. Okay, so why do we care about all these things? Well, to minimize disruption of the hydrogen bonding network of water. And another way to state it is, that is, to minimize the loss of strong favorable hydrogen bonds. The loss of strong favorable H bonds. Water molecules form a structured cage around the nonpolar species. Which in our case are nonpolar residues. Okay, and so can you answer in the chat whether you think the addition of a nonpolar solute molecule and this formation of a structured cage of water molecules would be entropically favorable or disfavorable? You can just type favorable or disfavorable in the chat, I guess. So I'll wait for like a few moments for someone to write in the chat. Okay, we have 2 answers so far, and they say the opposite thing. Does anyone else have any thoughts? Anyone else want to put in their 2 cents? Okay, we have one more participator, and they say not favorable. And so I would say that this is entropically disfavorable. And why is this the case? Well, when water is physically constrained in this cage, it can't rotate or move without disrupting its hydrogen bonding. And so another way to say this is that water is losing degrees of freedom. So this is entropically disfavorable. And so when hydrophobic residues interact with each other, it reduces the total surface area of non-polar stuff exposed to the aqueous environment. So interaction of hydrophobic residues with each other reduces their surface area exposed to water, which in turn decreases the disruption of the water hydrogen bonding network that we've been talking about. Oops, that's a bad end. And this in turn increases the entropy of the water. And of course, you know that increasing entropy is favorable. Okay. So I posted these notes and I'll post in the filled in ones. This is if you want more info. I don't think you need to read it. It was assigned to me when I was in this class and I thought it was helpful for me, but I don't know if you want to. Okay. It's very short, it's 1 page. Now I'm going to talk very quickly about membrane proteins. So here, you've seen this before. There's a polar head group and hydrophobic hydrocarbon tails on the phospholipids that make up the lipid bilayer. And the bilayer has a hydrophobic interior right here. And this hydrophobic interior has no available hydrogen bonding partners. And the consequence of that is that there's a large energetic penalty for entry of polar molecules. And the reason for that is that they would lose hydrogen bonds with water without compensatory hydrogen bonds forming. So net loss of hydrogen bonds. Okay, so how do transmembrane proteins deal with this problem? That is, how do we satisfy the hydrogen bonding requirements of the protein backbone in the hydrophobic environment of the interior of the membrane? Oh wow, it turned really dark while I was sitting here. And the answer we already talked about when we talked about secondary structure, and that's that hydrogen bonds form between different regions of the backbone. AKA secondary structure. Okay. And so the two kinds of secondary structure we talk the most about are alpha helices and beta sheets. And so here is a transmembrane alpha helix. And you can notice that these yellow residues, which depict hydrophobic residues, are in the membrane spanning region. And this hydrophobic region is about 30 angstrom. So if the rise per residue in an alpha helix is 1.5 angstroms, you can note that it takes 30 divided by 1.5 equals 20, oops, not angstroms, residues. It takes about 20 residues in an alpha helix to span the hydrophobic region. Okay, and Andrea already talked about why membrane alpha helices are more stable on their own, I think, as compared to isolated alpha helices in solution. And the reason is that an alpha helix in solution has competing hydrogen bonding partners. So if you think about that for a moment, what this means is that the denatured backbone can hydrogen bond with water. So the solution alpha helix, the alpha helix in solution, is less stable relative to its denatured form. Don't want that. Okay, and then finally, we touch on beta sheets. So you might notice when you look at the structure of beta sheet that there are all these unsatisfied hydrogen bonding partners on the edges of the beta sheet. And so that immediately signals to us that it can't traverse the membrane like this. Instead, what happens is the beta sheet rolls up into a barrel-like structure so that these ends with unsatisfied hydrogen bonding partners can touch each other and have hydrogen bonding interactions. And so this leads to a structure called a beta barrel, which can cross the membrane. Okay. And so finally, this is just a figure from your textbook showing you different types of transmembrane proteins and maybe attached globular regions. So you can read the figure description and take a look to see these different varieties. Oh, and finally, I just included the dimensions for your reference if you use these notes in the future and you need those numbers. Okay, so if you have any questions, please ask me in the chat and I will turn it over to Shivani because it is 6 p.m.
Hi guys, I'm just going to start recording.
Sorry, I'll stop my recording.